Welcome to Cell and Molecular Biology. Today, we're diving into one of the most fascinating and elegant mechanisms in molecular genetics, the Lac Operon. Imagine if your body kept producing digestive enzymes for food you haven't even eaten. It would be exhausting. Bacteria have solved this problem brilliantly. We're going to explore how they use a microscopic smart switch to conserve energy. A discovery so profound, it won a Nobel Prize and laid the foundation for modern biotechnology. To truly understand the Lac Operon, let us start with a simple analogy. Imagine you own a factory that makes a very specific tool designed only to crack open walnuts. If there are no walnuts around, it would be a massive waste of electricity, money, and materials to keep those walnut cracking machines running all day. Instead, you would install a smart sensor that only turns the machines on when a truckload of walnuts actually arrives at the factory doors. Bacteria face the exact same problem when deciding which enzymes to build for food digestion. They need a sensor. Now that we have our factory analogy in mind, let us look at the biological reality. In molecular biology, the Lac Operon is a classic example of an inducible gene regulatory system, famously studied in bacteria like Escherichia coli. But what exactly is an operon? Simply put, an operon is a cluster of genes that are transcribed together as a single long messenger RNA molecule. Instead of each gene having its own on and off switch, they are all controlled by one shared promoter region. This ensures that when the cell needs a specific metabolic pathway, all the necessary components are produced simultaneously. Building on the concept of an operon, let us examine the specific machinery of the Lac Operon. It contains 3 essential structural genes, each acting like a specialized worker in our factory. First, we have LAC-Z, which produces an enzyme called beta-galactosidase. This is the heavy lifter that actually breaks down the lactose sugar. Next is LAC-Y, which produces permease, a protein that acts like a doorway to bring lactose from the outside environment into the cell. Finally, LAC-A produces transacetylase, which assists in the overall metabolic process. Together, these 3 genes provide everything the bacterium needs to utilize lactose. So, how does the bacterium keep these genes turned off when there's no lactose around? This is where the repressor protein comes in. In the absence of lactose, this repressor protein binds tightly to a specific DNA sequence called the operator. Think of the operator as the physical switch on our factory floor. When the repressor is bound there, it acts as a massive physical roadblock. It completely blocks RNA polymerase, the enzyme responsible for reading the genes, from moving forward. This elegant mechanism ensures that the cell's precious resources are never wasted on unnecessary tasks. But what happens when our truckload of walnuts, or in this case, lactose, finally arrives? When lactose is present in the environment, a small amount enters the cell and is converted into a molecule called allolactose. Allolactose acts as the inducer. It binds directly to the repressor protein. This binding causes the repressor to physically change its shape, a process known as an allosteric shift. Because of this shape change, the repressor can no longer hold on to the DNA and is forced to let go. With the roadblock removed, RNA polymerase is free to transcribe the genes. Let us bring all these pieces together visually. On your screen, you can see the DNA strand laid out horizontally. Notice the promoter region, where the gray RNA polymerase is waiting to start its job. Right next to it is the operator, the critical switch. You can see the red Lac repressor protein. Crucially, notice the small green allolactose molecules binding to the repressor. This interaction is causing the repressor to detach from the operator. Because the repressor is moving out of the way, the RNA polymerase can now slide down the DNA to read the LAC-Z, LAC-Y, and LAC-A genes. Having seen how the mechanism works, you might wonder why this specific system is so famous. Understanding the Lac Operon is absolutely fundamental because it was the very first mechanism of gene regulation ever discovered. This groundbreaking work earned François Jacob and Jacques Monod the Nobel Prize in 1965. But it is not just history. In the real world, scientists use the Lac promoter as a biological switch in genetic engineering. By adding a lactose-like chemical to a bacterial growth tank, we can force bacteria to produce life-saving human medicines like insulin. Modern biopharmaceuticals rely entirely on this foundational concept. Now that we understand the biological mechanism, let us look at it through the lens of mathematics. The operon is not just a simple on or off switch, it has a graded response based on inducer concentration. We model this using a modified Hill equation. V represents the actual rate of transcription. Vmax is the absolute maximum speed the operon can achieve. The concentration of our inducer, allolactose, is represented by I in brackets. Kd is the dissociation constant, showing how much inducer is needed to reach half maximum speed. The exponent n is the Hill coefficient, representing how multiple inducer molecules cooperate. Finally, V basal is the tiny background leakage. As inducer concentration increases, the transcription rate shoots up toward Vmax. Let us apply this formula to a concrete example. Imagine a bacterium is suddenly placed in a glass of milk. We are given a maximum rate of 100, a basal rate of 1, a dissociation constant of 10, a Hill coefficient of 2, and an inducer concentration of 30 micromolar. We want to find the actual transcription rate. First, we substitute all these known values into our Hill equation. We square the inducer concentration of 30 to get 900 and square the constant of 10 to get 100. This simplifies our fraction to 900 divided by 1,000, or 0.90. Multiplying by 100 and adding the basal rate gives us 91. Physically, this means the operon is operating at 90% capacity to handle the sudden influx of food. Having calculated the transcription rate, let us shift our perspective to the repressor protein itself. Even when there is no lactose, the operon is not perfectly sealed off. We can calculate the exact probability that the operator is blocked using the fractional occupancy formula. P bound represents the fraction of time the DNA operator is occupied by the repressor. R, in brackets, is the concentration of the active repressor protein floating in the cell. Kd is the dissociation constant for the repressor-operator complex. If the repressor concentration increases, the probability of binding approaches 100%. If it decreases, the operator spends more time unbound, leading to basal leakage. Let us see this fractional occupancy formula in action. Suppose the concentration of the active repressor protein in the cell is 40 nanomolar, and the dissociation constant is 10 nanomolar. We want to find the probability that the operator is blocked when no lactose is present. We substitute 40 for the repressor concentration in both the numerator and denominator, and plug in 10 for the dissociation constant. This gives us a ratio of 40 divided by 50. The final answer is 0.80. This means there is an 80% probability the operator is bound. Physically, this explains why 20% of the time the operator is free, allowing a tiny basal level of transcription, so the cell always has a few permease doorways ready. We have covered a lot of ground today, so let us summarize the key takeaways. First, the Lac Operon is a brilliant evolutionary adaptation, acting as a smart switch to conserve cellular energy. Second, it relies on a physical roadblock, the repressor protein, which stops transcription when food is absent. Third, the inducer molecule, allolactose, removes this roadblock when lactose is available. Fourth, we saw how mathematical kinetics, like the Hill equation, perfectly predict these biological responses, including crucial basal leakage. You now understand the absolute foundation of modern molecular genetics. Great job!